Right, good morning, good day, everyone. Welcome to Bioscience Network YouTube channel. And today we are going to continue in our MOE series. And today we are going to start docking. We are going to learn how to prepare our proteins for docking. So, in the previous video, we saw how we can create an MOE database file that we can use to set all our ligands that we'll be using for docking. So today we are starting with docking proper. But first off, we need to clean our protein. So let's get into it and open our working directory. So I have it in my downloads and I think it's here. Yes. So I want to use this. This is where I want to use as my working directory. So like in the last video, I changed the working directory here, and then I open this, and this is the PDB. So it's opening as PDB. Yes. Also, let's see, before I open this, let's still check some other things here. Although it's I don't think it's as updated as possible, you can still load protein database here, but you have to be online to do so. So you can type the code for it and then run it. Um, it fetches the PDB file for you and you download it. It also does from Silo. So, but that's not what we want. So I'm just going to use from my working directory, and I open my receptor. So it asks you most of the time the symmetry. You want to load it as models or as entries. You can just leave it as it is. But for me, I prefer either loading it as non or as biological or biomolecule assembly. So once you open it as BA, it applies biomes transforms to create the biomolecule. So if you don't really understand how you want it to, whether you wanted to use it as an asymmetric unit, you can just leave it as none. Click OK, and then it loads your PDB file here. I do not have my mouse with me, so I'm just going to use my trackpad like we did in the last video, and we use here to rotate. So we have this in our screen. Now we can check the sequence here, and then we can clean our protein here now. Yes, we can clean our protein. I think yes, first things first, we can remove water. Once we come to this sequence, it shows us everything. And here I can write control click on these chains, select them, and then for everyone I've selected, I can delete them. So these three chains and water atoms, I'm removing all of them. I don't need them. Now I'm seeing that uh it came with a co-crystallized ligand, and I believe we can make use of it. Yes, first and foremost, even as our docking site. Yes, as our docking site. And we can make use of it for docking. But there are other chains here. I'm seeing chain A, chain B, and chain C. And I see that chain A and chain C are the same thing or almost the same thing. Yes, chain A and chain C, they are not all the same. Seeing glycine as first, proline, leucine, glycine, serine, methionine, glutamate, and then alanine here. These are the differences. So we can see the difference between them. Now, if yours is not showing as 3 letters, you can come here, down here, down here, here. You can either toggle secondary structure annotation. Yeah, if you don't want that, you can change the residue number or the labeling. This, and if you don't want this, you can also come here using one letter. You can just use only one letter rather than the full three letters to save space. But I prefer mine as the three letters here. So I don't think I'm going to need chain B and chain C, since where my receptor is majorly where my active site will be is already on chain A. So I delete those other two chains. Good. So I have, let me reduce this, take this up. Good. Alright, so this is what I want here. Now once I rotate it, I think I can see where my ligand is, the co-crystallized ligand is, and it's here. Although if I want to see it clearly, I can come here down to this place. This you can reduce the size of the chain, and I want to make it bigger. Let's see if I can make it bigger. Good. And I can change the color if I want to. So using green. So here we are. Good. So first thing we are going to do now is to prepare our protein. So we come to compute, prepare, quick prep. That's the one I prefer using. You can use structure preparation, but it's if you want to set the parameters yourself. And I'm just dealing with this place. But for you to set everything by yourself, uh, if you really do not have information about the protein, for example, this structure preparation tells you the problems already there, and then the ones you would like to sort out, or the ones you can sort out, you sort them out. However, if it's quick prep, it just notices all the problem and then use preset structures to preset structures to work on them and then prepare the protein. So you can leave everything here as default, or you can change any one of them that you want, and then you run quick prep. So quick prep starts here. You see it does a little bit of homology modeling and sees places that are not really complete and tries to complete them. So wait for it while it does that. Now it's correcting library issues, charge issues. Okay, now it's protonating it. Good. Giving it proper energy, both the ligand, the co-crystallized ligand, and on the receptor. Usually it takes a little bit of time, but it doesn't take too long. And once it is done, you are going to see everything here is out. So we are done with preparation. And the next thing we have to do is to get our active sites. Now, there are two ways we can do so. Yes, there are two ways we can do so. One, we can, if we have a known active site where a co-crystallized ligand is already, for example, where my uh, co-crystallized LZ9, that's the um, that's the co-crystallized ligand used here, LZ9. This is where it is. We can just use this to dock, where we can go and dock directly. Or if we don't, we can come to compute and then site finder, and then we apply, and it finds the basic sites, the active sites, and possible ligand binding sites that we can use for docking. For example, I think it has found them, and out of everything, we have this possible ligand binding site. The best place a ligand if it's in this receptor, it will prefer to bind here. So if this PLB score here is really high, and it shows that yes, this place is a better place to bind. So this is how the active site looks like. So since we have this as the active site, we can select the residues in this active site and then close. Two options. Now, if I want to dock, I've selected it now. Most of the times, people do not like leaving their co-crystallized ligand if they want to dock, if they want to use it for docking. But for me, I can leave it there, select that ligand as a place for my active site, use it as a place for my active site, and then use it for docking. Then I remove it while docking starts so that it does not prevent my ligands from binding. So this is how you can prepare your ligand, your receptor for docking. And so in our next video, we are going to start docking properly. Thank you for watching.